Hello friends, welcome to Sam Biology class. Friends, today we shall discuss about the structure of antibodies. Antibodies, also known as immunoglobulins, are circulating proteins. They are produced in vertebrates in response to exposure to antigens. They are incredibly diverse. They are also specific in their ability to recognize foreign molecular structures. Antibodies can exist in two forms, membrane bound and secreted. Membrane bound antibodies are present on the surface of B lymphocytes where they act as antigen receptors. Secreted antibodies reside in the circulation, in tissues, and also at mucosal sites where they prevent the entry and spread of pathogens. B lymphocytes are the only cells that produce antibodies. All antibody molecules share the basic structural characteristic but display tremendous variability in the regions that bind antigens. It is because of the variability in these antigen binding regions. Different antibody molecules bind a tremendous number of structurally diverse antigens. An antibody molecule has a symmetric core structure consisting of two identical light chains and two identical heavy chains. The molecular weight of light chain is about 25,000 and that of heavy chain is 50,000. This figure shows the structure of an antibody. These green colored chains are light chains and this red colored chain and the black colored chains are heavy chains. The dotted lines represent disulfide bridges. Both light chain and heavy chain contain a series of repeating homologous units which are about 110 amino acid residues long. Each homologous unit folds independently in a globular motif called as Ig domain. This figure shows the structure of an Ig domain. Each Ig domain is composed of two layers of beta-pleated sheet connected by disulfide bridges. Each beta-pleated sheet contains 3 to 5 antiparallel polypeptide chains. Here you can see that these polypeptide chains are antiparallel. Red colored polypeptide chains represent one layer of beta-pleated sheet and the black colored polypeptide chains represent another layer of beta-pleated sheet, and these two layers are connected by a disulfide bridge. In a beta-pleated sheet, adjacent polypeptide chains are connected by short loop. It is the amino acid in some of these loops which are critical for antigen recognition. In this figure, this is an Ig domain and this is another Ig domain. Similarly, in the heavy chain also, these are the Ig domains. And this is the disulfide bridge which connect the two layers of beta-pleated sheet. So, in antibody molecule, both the heavy chain and the light chain contain a number of Ig domains. Both the light chain and heavy chain consist of amino terminal variable region that participate in antigen recognition and carboxyl terminal constant regions. The constant regions of the heavy chain mediate effector function. In this figure, V represents the variable region and C represents the constant region. The V region of heavy chain is composed of one Ig domain and the C region is composed of 3 Ig domains. Similarly, the light chain is composed of one variable domain and one constant Ig domains. The variable region of one heavy chain and the adjoining variable region of one light chain form an antigen binding site. because the core structural unit of each antibody molecule is composed of two heavy chains and two light chains. All antibody molecules will have at least two antigen binding sites. In this figure, this is an antigen binding site and that is another antigen binding site. Each antigen binding site is composed of a variable region of the heavy chain and the adjoining variable region of the light chain. The constant region domains are separate from the antigen binding sites. They do not participate in antigen recognition. The constant regions of the heavy chains interact with other effector molecules and cells of the immune system and mediate most of the biological functions of antibody molecules. In addition, the heavy chain exists in two forms which differ at their carboxyl terminal end. One form of heavy chain anchors the membrane-bound antibody in the plasma membrane of B lymphocytes, and the other form of heavy chain is secreted by the B lymphocytes after it is associated with the immunoglobulin light chain. The constant region of the light chain do not involve in effector function and are not directly attached with the plasma membrane of B lymphocytes. In an antibody molecule, the light chain and the heavy chain are covalently linked by disulfide bond formed between cysteine residues in the carboxyl terminus of the light chain and the CH1 domain of the heavy chain. In addition to this, non-covalent interactions between Bl and Bh domain and between Cl and CH1 domain also contribute to the association. In an antibody molecule, the heavy chains are also covalently linked by disulfide bonds. In IgG antibody, these disulfide bonds are formed between cysteine residues in the CH2 domain, which are closer to the region known as hinge. In other isotypes, the disulfide bonds may be present in different locations. In addition to this, non-covalent interactions between the CH3 domain also contribute to the heavy chain pairing. Antibody molecules are the primary mediators of humoral immunity against all class of microbes. The association between the chains and the functions of different regions of antibody molecules were first deduced from experiments conducted by Rodney Porter, in which rapid IgG molecules were cleaved by proteolytic enzyme into fragments with distinct structural and functional properties. In rapid IgG molecules, the unfolded hinge region is the segment most susceptible to proteolytic cleavage. So when using the proteolytic enzyme papain, cleavage occur at the hinge region that resulted in the formation of three pieces. These are the three pieces of fragments that are formed due to the proteolytic cleavage of rapid IgG molecules by the enzyme papain. Out of three pieces, two pieces are identical to each other and each piece is composed of a complete light chain and VH and CH1 domain of the heavy chain. These two pieces retain the ability to bind with antigen. Because of this, these fragments are called as FAB, in which F stands for fragment and AB stands for antigen binding. And the third piece is composed of two identical disulfide linked peptide fragments containing CH2 and CH3 domain of heavy chains. This fragment has the propensity to self-associate and to crystallize in a lattice. Therefore, These fragments are named as FC. F stand for fragment and C stand for crystallizable. In contrast to this, when rapid IgG molecules are cleaved by the proteolytic enzyme pepsin, cleavage occurs distal to the hinge region that results in the formation of a fragment in which two antigen binding sites are present. Because this fragment has two antigen binding sites in which there is one complete light chain and there is the heavy chain fragment which contain VH and CH1 domain of heavy chain. This fragment is called as FAB2. So friends, with the help of these experiments, scientists became able to know about the association between the chains and to study the functions of different fragments in antibody molecules. So friends, this is all about the structure of antibodies. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please share your feedback. Thank you very much.